Today, we are going to talk about genomic surveillance. Now, that one alone is already now throwing you off. Genomic surveillance. Creating awareness on the importance of genomic surveillance in response to outbreaks. And the person who's joining us is the Bioinformatics lead at the KEMRI-Wellcome Trust Research Program, Dr. George Githinji. Dr. Good morning.
Good morning.
How are you?
I'm fine, thank you.
Welcome to the hot seat of the Situation Room.
Oh, thank you very much. It's lovely to be here.
Very good.
Thank you.
Early in the morning when you talked about this, we said, this guy is going to come and tell us about, the 666 on the forehead. And this is how we do the surveillance.
We do in science.
Related matters.
And such others, right? Well, we don't know. You will tell us what is bioinformatics, what is genomic surveillance, and all that. But before that, City has the day's proverb. He'll give you the proverb from which country this week?
DRC.
Very good.
An interesting country, this one. It has many firsts.
Many firsts.
Oh, yes, it does. It does. One, it is one of the very few countries, it's the only country in Africa that was once owned by an individual. Okay. The King of Belgium. Okay. It has the world's deepest river. Now, you may not think so, but Congo River is the world's deepest river.
Right.
Okay. This one is the third largest river by discharge also, given the distributaries and whatever else moves around it.
Third largest river by discharge.
Yes.
In the world.
Yeah. You know that river, when you look at it, it's actually, your idea of a river is something whose boundaries you can easily see. That it is huge.
This is a lake.
Yes, it is. It is a moving lake. Anyway. That is DRC.
Very good.
Yes, and its qualities. The rain does not fear the authority of a king.
The rain does not fear the authority of a king. Dr. Githinji, what's your interpretation of this proverb?
The rain does not fear the authority of the king. That's quite a deep one.
And it comes from the country with the deepest river.
Oh, indeed, isn't it? Well, yeah, no pun intended. I think it just sort of looks at our inabilities to control certain events, in the sense that you could be or hold immense power over people, but you can't hold that against the world and natural phenomena. And as such, it'll beat you as much as it'll beat a poor man, even though you're the king.
You put it very well, thank you.
Kabisa?
Kabisa.
Very good. Very good.
Nature is a great equalizer.
Yeah, indeed.
Yeah? It brings all of you together.
Same.
Okay.
Yeah.
Dr. what is bioinformatics?
Bioinformatics, we could sort of, look at the word itself. We have bio and we have informatics, and often in biology we generate a lot of data, a copious amount of data. When you're doing your experiments, when you're looking at specific things, and often you can't go through those data manually, and therefore you need a computer to help you do that. You need proprietary software in some instances, but you would need a dedicated informatics tool. And the people who develop those tools, the people who work with those tools, apply them, in as far as our understanding of biomedical research data, are bioinformaticians. And they tend to be multidisciplinary, and therefore you'll have your biology. For example, my basic undergraduate degree is in biomedical sciences, which in itself is a multidisciplinary degree, comprises of biochemistry and genetics, human anatomy, and understanding epidemiology. And then there is the computer science aspect of it, which you need to know how to look at data, algorithms, for example, that basically, recipes for, accomplishing a task by a computer. And bringing together those fields, including statistics, makes for this sort of a career in bioinformatics.
So you are all those things? You're a biomedical scientist,
yes.
You're a computer scientist, and you're a statistician.
Yes.
Dr. When you wanted to do this course, were there not simpler courses that you
It depends on what you define as simpler, because in my understanding, nothing is simple when you really look at it.
You could have just, for instance, studied biology just
alone,
and then gone and done media studies alone, and then done computer packages alone.
Which we all did back then. So yeah, you could do that, but then the problem that you want to tackle,
yes,
cannot be tackled by just that subject by itself, and therefore it calls for bringing in other disciplines.
What was your journey? What interested you into one into the next? For example, in your biomedical sciences, is it where you landed because of your marks, or did you actually want to end up there as your undergrad studies?
I actually wanted to land there.
Why?
Because I was keen on research. So I've been keen on research from high school. And I looked through the courses, and initially I thought, well, let me look at chemical engineering. And then I looked at it again. I was like, okay, this is interesting. But the cutoff, at that time was extremely high. And the class limits was very, very, very few individuals. And it was only at Moy University back then. So I looked at the odds and realized, well, I don't think I want to take this path. So I went back to JAB, as it were, the Joint Admissions Board, and I changed my course after looking through, the internet back then and realizing there's a field called biomedical sciences that takes bits of everything. And it was a relatively new course at the Edgerton University. And I went and applied, and I scored the mark. I got admitted. Along the way, I was always fascinated by computing. So right out of high school, I had done a programming course. Back then, we used to do Pascal and Basic and Visual Basic. And I started wondering, how do you get these together? And I realized that there is a room for it. And I just went on in it. And I did lots of it, self-learning, and just about when I was graduating, there were lots of workshops, opportunities were opening up. And it was, as a field, was already quite advanced in the West. And therefore, by, attending these workshops, getting exposed, having opportunities to, to collaborate, to go to the US and other places, I acquired most of these skills.
What has the, combined force really of all of this research studying, then allowed you to do in the fields for which they're important? Let's say in Kenya today, what has that allowed to come about as a result of your research?
That's a good one. And I think it speaks to whether have had impact in what we've done. And I have to say that I feel very privileged to work at the institution where I work, and with the colleagues that I've been able to work with. So a lot of the opportunities that I've had to contribute to, particularly around bioinformatics, data analysis, pertaining to respiratory pathogens, for example. I think that's where I've made a lot of impact, just looking at the opportunities that are there, were there to, for example, establish the platforms that we've established for genomics at KEMRI-Wellcome Trust Research Program and KEMRI overall.
Okay.
Yeah.
So genomics, I know we mentioned it before. Can we go into that a little bit to figure out, okay, so what is this thing? And what does it mean exactly, and how does it help in terms of medical research and, the possible conditions that Kenyans or others suffer from?
That's an interesting one. So we can look at the term again, and we can take a step back and understand what is really are we trying to do. So when you think about pathogens, they are either viruses, bacteria, or fungi, and at the base they have genetic material, and genomics overall is the study of that genetic material. So what we're going to do is that we're going to find that genetic material, and we're going to understand its makeup. And we're going to exploit opportunities in that genetic makeup to better understand what it is that we're dealing with. And that pertains to all living matter that has a genetic makeup. Viruses are interesting in particular because they only have that, and they then hijack other cells to make copies of themselves. I mean that in itself is very fascinating and very nerdy, which attracts me.
When you say this, you're getting the genomic material.
Yeah.
What's that? What are you getting?
So what we're getting, I should have broken down, is DNA. Or RNA, and in simpler terms is the building blocks of life. All of us have an alphabet or a code, if you like, and that code is made up of 4 bases: adenine, thymine, uracil, and cytosine. And those chemicals, those 4 bases, ACGT, encode blocks of life. They go through complex processes in our body, in our cells, to make proteins and so on and so forth. And if we understand that genetic makeup, we can do quite a lot.
Is it different from person to person, or from organism to organism?
It is not that different. So let's get this clear. So one, there is that what pertains to pathogens, which is, well, if you like, I don't want to use the word simple, and this, when it pertains to higher order organisms like humans and other vertebrates, it is not that different. The base copy is the same, but there are mutations or changes that makes us different. And understanding those changes, we can understand a lot. We can understand, for example, how we metabolize certain drugs, for example. We can understand why we look the way we look. And more important is that we can look back in the past and ask the question, where did we come from? Which is a fascinating topic by itself.
So when you're in a lab, just take us through practically, as simply as possible. Now take yourself back to Dr. Githinji when he was just Githinji in Form 2, all right?
Yeah.
What is it that you're doing? Are you in a lab looking through a microscope and looking, and what are you seeing something? Are you using a needle to extract something? When you say that you're getting the genetic material, what are you doing?
We are taking our pathogens of interest, or our cells of interest, and we are shaking them, we are perturbing them, and we extracting the genetic material, which is the DNA. You're using different processes and steps, and once we have that, we then going to take it through an instrument called a sequencer. And through that, we are going to translate those chemicals to an alphabet comprising of A, C, G, T, G, J, A, and so on and so forth.
Right.
And that is what we are going to work with moving forward.
Okay. So what does it help you do?
What it helps us to do is quite a lot, depending on where it comes from. And I'll mostly restrict myself to that genetic material that comes from pathogens, infectious pathogens.
From disease?
That cause disease.
Okay.
Yes.
All right.
Yes. So the pathogen is the
disease-causing agent.
Exactly.
Thank
there
you.
we go.
All
There we go. We're on it now. So that is what we're going to restrict ourselves to. And what we really going to do once we get that genetic material, we're going to ask us a couple of questions. One of them is if one of us was, sneezing, for example, or had an ailment, and we all have information that we were together somewhere, and we all started sneezing out there, and we provided a sample, and we extracted the genetic, the disease-causing pathogen. It probably could be a virus, it could be a bacteria. We can then look at the genetic makeup of those pathogens and ask ourselves, is it identical? Is it similar? And if there are big differences, then it is likely that, one, if there are no differences, and we have information that at some point, we were, in a room together, then we could argue that it came from one of us. One of us, infected the
one
other.
source.
Yes.
And then it distributed.
And if one of us, for example, or all of us had very different genetic material, or those pathogens had very different genetic material, then it's very unlikely that we got infected from one individual. It's very likely that we got it elsewhere. And that information is very important when it comes down to... Yeah.
Can you repeat what you just said? If you all have it, if you're all sneezing, for example, and you're able to now, you've taken the makeup of this pathogen, because you've done a sample from everybody, and it is similar.
Yes.
Then it is...
All identical.
Or identical.
Yeah.
It is unlikely that you got it from the same...
If it is identical, you likely got it
from
the
same place.
Yes, okay. Only if we have information that
all of you were together.
You all went to a concert on Friday, for example.
Yeah, exactly. Like we were all together at an event. And either we didn't have it or one of us had it. That is very important because the added information, genomics by itself, the sequence data is not very informative if we don't have other information, on it. And the other important thing is how soon or how quick do we get it. Yes.
You guys were together yesterday in the morning, but by yesterday 4 p.m., all of you were sneezing. Right?
Yeah. That's a simpler version of it.
Is this so now genomic sequencing, such a fancy term, is this the same idea that is behind tracing when it comes to diseases?
Patient zero,
COVID-19, Mpox or Ebola?
Yes, indeed. It's having that information in addition to other information about the diseased people. Where did they come from? What was their age? What symptoms did they have? And a very clean or clear line list in terms of, that's a big term, information about the collection or the infectious patterns, for example. So that can really help us.
Then the other side that you said was it's possible that all of you are sneezing, but when Dr. Githinji goes to the lab, he'll find that these pathogens are not similar.
Yes.
And so now here you'll have an issue.
You have an issue there because then if we find them to be very different, it implies that it is each of us have their own pathogen that they came in with from somewhere else.
Where?
And so that could be, I imagine that would be potentially worse.
Yes, because it means that there is a lot going out there
that you do not know.
That we do not know.
So you need now to start spreading.
Now we need to
fear to get more information.
Yes, we need to have more information.
CT, where were you yesterday? Who are you with? Eric, where are you? Who are you with? And so on and so forth.
And now you can imagine the situation where, as a public health official, as a government or a health agent, getting that information as soon as possible or in near real time. It can really impact on the control measures that you put in.
I just want to ask before we go. So we would ask then, I know you talk about control measures, but then why else would it be extremely important to do this? Because folks are going to be like, okay, so what? You're sneezing, there's a disease burden, we know it's there, treat, move on. Why is it important to sequence all of this and to do that research when there's an outbreak of whatever it may be?
Now, we're going to put a little bit of another scenario. Let's say we're not just sneezing, but it's a hemorrhagic fever. And now we've upped the stakes. Fatalities are happening.
People are dying.
People are dying. And people are panicking. And therefore then having that information about what is causing that becomes really, really important. And also understanding the transmission patterns. And whether we actually know what it is that is causing that. Often, most of these ailments have overlapping symptoms. So if you bleeding, if you sneezing, if you coughing, you diarrhea, it could be one of a myriad things, like people have those sort of symptoms. If you have a common cold, which, if you're an adult, maybe that's not such a big thing. But if you have something else, then that becomes really important to understand.
You have a dry cough, you're short of breath, you're... and all those things.
Yeah. So it becomes really, really important to get that information as soon as possible. But more important is the potential to understand the spread and which demographics or which populations are getting affected, and whether we already know what it is, so that then we can, start implementing measures.
Okay. Genomic surveillance. Siti, so far, have you seen how genomic surveillance, for example, helped us to start saying this was patient 0 for COVID-19? Or this is how COVID-19 is spreading. Let's shut down this part of Nairobi. Let's go to Kawangware and do a lot more tests in Kawangware as opposed to a lot more tests in Jericho. Why the thinking was like that?
I heard what he said.
You heard?
I'm still trying to process what he said to see if I understand it.
Daktari, then continue. For example, using the case of COVID.
Yep.
Now we know we have gone through COVID, all of us as a country.
Yes.
Now show us how genomic surveillance was important during that time.
Very, very, very important. One, let's take a step back before we knew that it was COVID, because COVID is the disease, and the causative agent is a virus called SARS-CoV-2. And before we knew about that, or health authorities knew about it, people were simply presenting with pneumonia and lots of fatalities initially in China. And it was a new ailment that people didn't quite understand very well. So the ability to, one, understand that this is a new novel pathogen involved genetic sequencing and genomics. So they took a few samples from the individuals, they looked at the genetic makeup, and they realized that this thing looks like something we've seen before, and that's SARS virus, which came in earlier. But it tends to happen that these are coronaviruses that are actually prevalent, a lot more prevalent in bats and in within communities. But this was a novel one. So right there is the impact and utility of genomics. The other one is our understanding.
Hold on.
All right.
Does it mean that as the researchers in the labs were then getting these samples from the patients, they also still had the other samples from the previous SARS outbreaks, and they were comparing?
Yes, because this is a set of viruses that are known to cause common cold-like illnesses. And once you take the genetic makeup, you can look against a database of all pathogens that have been sequenced to date and ask the question, is this something that is already there, or it's similar to what?
Have we seen it before?
Yeah, have we seen this before? Okay. And if you do that, then you realize that it can narrow down to a few individuals of something that has already been done before. And when it's novel, you realize that, this is something that doesn't...
There's something a bit different
absolutely
from the others.
Yes.
What would that be? For example, in the case of COVID, because we have had SARS viruses before. We've had, these things have been there before. What was what was the new thing about COVID?
One, because again, on the SARS-CoV-2, looking at the genetic makeup, it was quite different in terms of you remember what we talked about, the ACTGs? It was quite different. It had novel amino acids that are not in other coronaviruses, and that's why it became the novel coronavirus, if you remember.
Right.
We call it new.
Yes.
Before then the WHO coined the term SARS-CoV-2. The 2 means is the second one. Although there are like several of them.
It is new ship.
Yes. The other important thing is, and this led to a bit of a controversy. Where did it come from?
Yeah.
There was all this controversy about a lab origin. Yeah. There was the controversy about animal origin.
Wet markets.
And when you look at the genetic makeup again, you realize that this is not something that was baked in the lab. This is something that was already there, and there was a spillover from animal reservoirs. And you remember what I told you about other information? Again, going back and looking at how and where did the individuals that were, first initially infected, where are they coming from? What did they interact with? Did they handle animals? Oh, yes, they did. Mm-hmm. It's in a massive market where live animals are getting sold. And do we find these viruses in these animals? Yes, we do. So it's a spillover.
Doctor, the process that we're describing here takes place in a lab. Sequencing, determining, understanding, that entire process. So if that entire process takes place in a lab, what is it that stops it? Now, I'm being a conspiracy theorist here, forgive me, but what stops one from tinkering? When one has the knowledge that you have, why don't I tinker with this and tinker with that and then see what happens? Because I already understand their makeup. I know what they do. So let me see now, if I mix this and this, what will happen? Will they die off? Will they create a hydra? I'm simply saying...
Yeah.
It's true.
Yeah, that's a real concern and a very important one. And that's why in research we have research ethics. And you are bound by research ethics. And there are colleagues that look at what you're doing and they can criticize positively and tell you that, looking at the data that you've provided us and what you've done, we don't think that the assumptions you're making are real. And then you go back to the drawing board. And then your aim is to convince these colleagues of yours, the peers, it's called peer review, that this is actually something that, holds water.
Or the reverse could happen, whereby at the end of the day, if we find that actually, you know what, this was not a natural thing that happened, maybe somebody actually did cook something in the lab.
Yes.
And that's why folks could actually then be on the chopping block in the future if your original information was not true, right?
Yes.
Okay.
Yes, and it carries serious implications. Internationally. I mean, there are laws that govern, what we do. There are laws that govern, as I mentioned, human ethics and so on. And therefore, it's very important that you get peer review and that positive criticism.
Can I ask this question?
Yeah, sure.
You see, one of the things that I have found puzzling over time is that every time there is an outbreak of sorts, for some strange reason, we always identify the areas of DRC as where it emanated from. Okay, now, if I were to look at the human-animal interactions in that region, and even the diet that involves game and wild animals, if we were to take the argument that we used regarding COVID, and how it is that wet markets where there were animals and so forth, and you now take it to this setting where the human interaction with animals has to do with them eating it and perhaps not preserving them enough, or perhaps not doing whatever it is that you need to do to ensure that any pathogen that may be harmful to you is extinguished. Is there a remote possibility that indeed this occurrence that seems to be more, in my mind, than coincidental could arise simply because of the factors that I've mentioned?
Yes, that's a real concern, and indeed, eating of game, particularly if it's not prepared, in a particular way, to make it palatable, or should cooked. I use that word, or cooked? But even when it is, it's very important that to be aware that, that could be a way through which, a spillover can happen. But most spillovers will happen mostly through, contact with these, with the material. So it could be blood, it could be, respiratory. And often it involves a specific, a change within the parasite. So for example, I'll give you an example. Let's look at MERS coronavirus, which is of same family, but it tends to infect camel. And it spreads among camels, but you, as an individual, as a human, you can get infected as a result of close proximity with camel.
Kissing a camel.
And you remember we had a major outbreak Well, I'm trying to get that picture in my mind. That it's quite funny. Yeah.
And you're just thinking,
I can't avoid
will not sneeze into your face.
Or sneezing onto your face. You know.
Unless you put your face next to it.
Yeah.
Yes.
Yeah. Mm-hmm. So it turns out that a human can get infected, but in terms of transmission, you don't have human-to-human transmission because you've not, the virus has not made that jump, that critical mutation that is needed to then, have human-to-human transmission.
My mind is thinking, if we can have a situation where pathogens that exist in animals can affect and infect us, what about the other way around? Can we affect and infect animals as well?
Yes, we do. And if you again going back to COVID, there was a lot of game sampling that happened, and indeed other animals, cats and felines, were affected because they were getting this from humans.
Oh, we gave COVID to cats.
Yes, we did. In some instances, in limited settings, we did. So the other way can happen, which brings us to a very important thing about encroachment and wildlife conflict and climate change that is going to cause or is causing
more and more
movement of people, mobility. As climate change exacerbates, we're going to see movement of people into areas, and the other way around, of vectors or animals, disease-causing agents in areas that they initially didn't, colonize or they didn't occupy. And that's a real concern.
I imagine that when it comes to genomic surveillance, you don't wait for outbreaks. That once you've seen, for example, the common cold, you constantly keep monitoring the cold to see whether it's mutating, to see whether it's, changing in terms of how it even responds to the treatment methods that we put into it. Is that correct?
Yes. So it is very important. I think what you're talking about is doing surveillance. And we have specific studies. So, for example, in KEMRI-Kilifi, we carry out a lot of surveillance at multiple levels, within health facilities, within inpatient, outpatients, and, dedicated studies that are looking at particular populations, and that has been going on for a long time, and we have a very great platform for engagement with our local communities, and that ensures that there is consistent surveillance for multiple pathogens.
Have you been able to detect something before it became a major issue? For example, through surveillance, you start noticing a change in the pathogens, that then you raise the alarm and say, this thing is starting to become a serious issue.
Yes, a lot of times. Examples, we can go back historically and look at malaria. So if you remember, there was a time, chloroquine was the drug of choice for malaria. And a lot of resistance came around. So again, looking at, the specific segments of the parasite, you can start to look at where the drug is impacting what we call pressure, or selective pressure, and therefore it making the parasite behave differently, and therefore it's able to overcome whatever drug pressure that you're giving it, and therefore it changes. But then what that does to the parasite is it puts it into sort of a corner of evolution that it doesn't want to. So when you lift the pressure, as it happened when chloroquine was discontinued, if you go back now in a lot of countries and other areas, you realize that the parasite bounced back, and now they became susceptible to chloroquine.
Whoa.
So there is quite good genetic data that that can help us in that. The other one is, again, just jumping back, and we can look at Ebola, we can look at SARS-CoV, our understanding of the so-called variants of concern, and we've also been able to pick up outbreaks, at the very early onset, simply by tracking individuals that are presenting with specific symptoms within our surveillance platforms, and able then to say, well, this is something that we think, is of concern. Yeah.
So it does make the case then for research, because sometimes we think, okay, there's a lot of things happening in the back end, and we may not see that we won't see the results. We just know, okay, you go to hospital, you get treated, you're seen by a doctor, they make diagnosis, you get treated, you go home, you get better, right?
Yeah.
We don't see what happens in the back end.
Yeah.
But as we see all of this, then it starts to you connect the dots and you start to understand.
Yeah.
Then why it's important to do it, and then why the call continues to be on governments to actually invest in medical research, isn't it?
Yeah, that's a very important point you brought there, because what you really want is a lot of centers of excellence, and you want government to invest in research. Our bastions and pillars of research are often universities that are hugely underfunded. If you go to any developed country, that is where basic research happens, because there's research that happens, and this is usually the sort of the frontier pushing type of research, what we call basic research. It's not something that you're going to go to a partner and tell them, oh, by the way, I really want to understand the signaling of my particular molecule of interest. You'll be asked, and what is the impact? And if there's none, maybe they're not interested. But that can turn out to be the most important thing that you want to do. And often that type of research is driven by universities, which unfortunately there's very limited case of in this country. And I would like to call, for increased funding, particularly for universities and research, and the expansion of research centers. A lot, again, of our research is done collaboratively with our partners and also within government. But you could imagine if the government segment was expanded, not by half, not by quarter, not by a 15th. I'm just asking by a 5th. That can make a huge impact. And then we can start, moving forward.
Scale it up.
Yeah, we can scale it up.
Use funding notwithstanding, is there sufficient interest that is generated by our universities so that research is something that is understood to be at the very, very fulcrum of an institution of learning like a university? Do we have enough interest?
I think we do, and by definition, universities are intellectual places, aren't they?
Supposed to be.
It's supposed to be.
Yes.
But
are they?
And that's debatable in some instances.
Yes.
But I still want to believe that at the core mandate of a university, intellectual excellence and basic research.
Citadel of knowledge.
The citadel of knowledge. That's what you want our universities to be. And you want them to, have the support, and then, we can carry out our mandate. So.
Dr. Githinji, how many people do you lead as the bioinformatics lead?
That is, so my position, cuts across the program, and supporting multiple groups, which in themselves are independent. Right. They are carrying out their specific research, across the genomic spectrum, across multiple pathogens. But then we have to have sort of, core utilities, for example, our high performance cluster, our genomics, lab platform. And then I have a lead, a small team of individuals, research officers, postdoc, and assistant research officers, technicians, to carry out, particular investigations.
Someone who's listening to this and they feel I want to learn more. I want to get interest in this. I want to end up in university or my postgrad and all, getting into this kind of research. How do they reach out to KEMRI-Wellcome Trust?
Yeah, so KEMRI-Wellcome Trust runs a couple of programs, and there's an excellent platform called the Ideal platform, and we have a dedicated training department, and they engage and onboard people at multiple levels, right from school leavers to university interns. We run together with the Pwani University a very successful program, the postgraduate diploma course at Pwani University. And then there are, opportunities for PhD fellowships and early postdoctoral fellowships. All these are often available through our training department and on the KEMRI website. So, and they're competitive. So, yeah.
It's good to know that we have such people in this country who've gone through, university here and who are doing all this to give back to our society. Thank you very much, Doc, for joining us.
Thank you very much.
Very inspirational.
Thank you very much.
Dr. George Githinji is the Bioinformatics lead at the KEMRI-Wellcome Trust Research Program. He's been telling us about genomic surveillance and the importance that it plays in response to outbreaks.
This is the Situation Room, the only way to start your day.